Hello, I'm Sabina Payalunga, Senior Director of Scientific Affairs at Celerion. And today I'm going to be discussing study designs and best practices for renal impairment PK trials, key takeaways from recent FDA guidance. In this webinar, we'll briefly review kidney function and physiology, how kidney function is measured, and design and operational considerations for renal impairment and dialysis studies. For those of you not familiar with Celerion, we are a full-service clinical research organization with clinical pharmacology focus. We specialize in healthy phase studies and clinical pharmacology studies to support drug labels, like renal impairment PK studies. In addition to clinical conduct, we offer regulatory and protocol writing services, data management, and bioanalysis. We work with large pharma and small to mid biotechs, and a good proportion of our work is with the small to mid biotech companies, where we've ranked quite highly over the past few years through the CRO Leadership Awards. Celerion is a global company with clinics in Lincoln and Phoenix in the US, as well as Belfast in the UK. And our bed capacity totals up to around 650 beds. We have bioanalytical labs in Lincoln and Zurich, Switzerland, as well as offices in Montreal and Vienna. Now to the topic at hand, let's get into kidney physiology. So the kidneys filter and excrete waste from the blood in the form of urine. They balance electrolytes and help maintain blood pressure and produce red blood cells. A nephron is a filtering unit where unfiltered blood enters the glomerulus, which filters small solutes. The tubules reabsorb glucose, amino acids, and electrolytes back into the bloodstream, while the collecting duct regulates the composition of tubular fluid and reabsorbs and secretes water and electrolytes. Now, chronic kidney disease affects approximately 1 in 7 people in the US. It's more common in adults, those older adults, those over 65 years of age, and more prevalent in women than men. Some of the risk factors for chronic kidney disease include diabetes, high blood pressure, heart disease, obesity, and a family history of CKD. Now, the disease itself is defined as having 3 months of abnormal kidney function based on cause, glomerular filtration rate, and albuminuria. The graphic here on the right shows the CKD staging risks and how it's stratified by glomerular filtration rate levels as well as albuminuria. And I wanted to point out that there are 6 stages to GFR, and they rank from high or normal, which is above 90 mils per min, to kidney failure, which is less than 15 mils per min. Now, GFR measures nephron plasma volume filtered per time during urine formation. It can be measured by direct and indirect methods. Inulin clearance is considered the gold standard, where inulin is continuously infused intravenously while timed urine samples are collected. Iohexol and radiolabeled markers can also be used, but these are expensive and time-consuming types of studies and trials. That's why we have indirect markers, which is a little bit more easy and handy to use. These indirect markers include creatinine and cystatin C. Phosphocreatine is metabolized in the muscle to generate creatinine at a constant rate, which is then filtered by the glomerulus. Cystatin C is a small protease inhibitor produced in nucleated cells, also freely filtered by the glomerulus. There are several formulas that have been developed using these 2 biomarkers to help estimate GFR. You might be familiar with creatinine clearance. This formula was first developed in 1976 by Cockcroft-Gault. Here, the formula applies serum creatinine, weight, age, and sex. Now, this formula does poorly estimate GFR because it's really designed as a model of creatinine clearance. It was first validated in a small cohort of participants using an outdated biosystem by today's standards. So in 1999, the modification of diet in renal disease was validated and published. This formula is developed to overcome the limitations of creatinine clearance. You'll see that there's no weight consideration, but it does apply a body surface area index. And I'll get into that a little bit more later in the talk. The formula is based on serum creatinine, age, sex, and does have a racial component. One limitation of this formula is that it underestimates GFR, especially in the high to normal ranges of GFR, mainly normal or mild impairments. So in 2009, the Chronic Kidney Disease Epidemiology Collaboration, or CKD-EPI, came out with a formula aimed at correcting some of the weaknesses of the MDRD. The formula is shown here. In 2012, they actually created and validated a formula using cystatin C, as well as a formula that applies both the combination of cystatin C and creatine. And then in 2021, a revised version of this creatinine formula was validated to remove the racial component. The European Kidney Function Consortium also developed and validated an equation that covers the full spectrum of age, ranging from children to adults. Their formula does apply a Q value, which is race dependent, and they also validated a cystatin C and combination formula in 2023. So that leaves us to today, where we have several kidney foundations and societies that have endorsed the use of both creatinine and cystatin C formulas to estimate GFR. And this is just a snippet from the KDIGO guidance, really pointing here that they say that if cystatin C is available, it should be combined with creatinine to estimate GFR. Now, there are many other formulas and equations available. However, the ones that we just covered here are the most widely applied in clinical trials. These two biomarkers, creatinine and cystatin C, are inversely related. So low creatinine or cystatin C levels tend to correlate with higher GFR, which is that normal function kidney range. When comparing these two biomarkers head-to-head, cystatin C has increased sensitivity for staging CKD, especially in that normal function mild impairment range. And you can see in the graph in pink, the creatinine levels don't really change much between 40 to 59 mils per min. And this range is termed the blind range. On the other hand, cystatin C in blue performs better across all CKD stages, avoiding the blind range. There are some limitations to these biomarkers. For instance, serum creatinine is dependent on muscle mass, so low or high muscle mass could over or underestimate the true GFR. It's also impacted by protein uptake. Creatine is found in meat and fish and is converted to creatinine during the cooking process. So non-meat eaters tend to have an overestimation of GFR. Creatinine also undergoes tubular secretion, which accounts for 20 to 40% of creatinine clearance. And then with cystatin C, it is affected by thyroid dysfunction and thyroid hormone treatment, as well as steroid treatment, which can lead to increased levels and underestimate the true GFR. There is also some debate and discussion about if cystatin C changes much under conditions of obesity, diabetes, and inflammation, and more study is needed here to confirm this. And then one other point I want to mention is that the cystatin C assay itself may not be readily available in clinical laboratories. And it also has a little bit of a higher cost associated with this analysis compared to creatinine. So that might have hindered its uptake as a common formula for estimating GFR. But nonetheless, given these limitations, formulas that actually apply both creatinine and cystatin C do perform better than the one biomarker alone. And that's why the societies are pushing to use both. When we were reviewing the formulas, you may have noticed that the eGFR was all indexed to 1.73 meters squared, and that's for a body surface area correction. Now, this value actually comes from the 1920s and is based off a small cohort of white American 25-year-old men. However, since then, the average weight and height of people in the US have increased. So indexing to 1.73 meters squared in participants with a higher BMI can actually underestimate GFR. That is why it's important to use absolute or de-index GFR. And to de-index, what you do is calculate an individual's body surface area, multiply it by the GFR value, and then divide by 1.73 meters squared. And this will give mils per min, or the absolute value. There are several formulas to calculate the body surface area, and the Dubois and Mostler are the most commonly used in clinical trials. So this is a really interesting prospective study. It's a recent study highlighting the importance of de-indexing, especially for patients with a higher BMI. Here, the authors compared the indexed and de-indexed eGFR values across 4 common equations: the EKFC, MDRD, and then the 2 CKD-EPI formulas. The participants were then grouped by their BMI. And the graph here shows the difference in eGFR after de-indexing. You'll note that the de-indexing resulted in higher eGFR scores, especially for those with a higher BMI. So then this in turn affects their CKD staging, meaning they would go into a lower stage where there is a less severe diagnosis. And this finding actually had major implications related to the type 2 diabetes medications they were on. Either they were given a lower dose than recommended or contraindicated completely. So, for example, patients were not given metformin if it was contraindicated based on their eGFR value indexed to 1.73 meters squared. The higher BMI group was most affected, and in the graph you could see anywhere from 3 to 14% of patients were inappropriately prescribed a diabetes medication based on their BMI and the formula that was applied. Now, a clinical pharmacology study is needed to determine the appropriate drug dosage for renal impaired patients. Kidney damage can lead to higher drug exposures and longer half-lives, resulting in slower drug excretion, as shown in the graph on the left with the time concentration curves. In addition, the buildup of body waste products can displace drugs from binding to plasma proteins and affect drug clearance. Both situations can potentially impact drug safety and efficacy, requiring either a lower dose or the drug to be contraindicated. Now, a renal impairment PK study is really a hybrid type study, pulling design features from both early phase and late phase clinical trials. In terms of early phase 1 study design aspects, here we're talking about a small number of patients with a single dose where precise PK time points are needed for sample collection. And typically this is over a 48 to 96 hour period. As a late stage clinical trial, it pulls aspects of a multi-site study where 2 to 4 sites are typically used with 1 to 2 patients enrolled at a time. The medical management of patients is a top concern, and then comorbidities and concurrent medication can be a major recruitment and study execution challenge. The FDA has issued draft guidance and more recently final guidance on how to conduct these renal impairment PK studies. And there's been some considerable changes to these drafts based on stakeholder feedback, really related to the worst case scenario. The draft guidance in 2010 defines the worst case scenario as end-stage renal disease patients not yet on dialysis. Now, at the same time, an advisory committee met and felt that this group of patients not yet on dialysis was not going to be feasible. So they recommended that dialysis patients during an off-dialysis days would be a better estimate of the worst case scenario, but also felt that patients with a GFR less than 30 would be sufficient. So 10 years later, the FDA revised the draft guidance and now defines the worst case scenario as kidney failure patients, which a value of less than 15 mils per min, or dialysis patients not on dialysis days. And then more recently, the final guidance that came out just last year actually combines the two groups. So we have severe renal impairment or kidney failure not on dialysis, representing the worst case scenario. This widens the term for this worst case scenario, granting more leeway for recruitment. So what else does the guidance say? Well, it covers topics such as when to run a renal impairment PK study, how to determine renal function, considerations for a full and reduced studies design, as well as dialysis studies, and then provides input on the PK, PD, as well as dosage and label recommendations. The FDA recommends that a dedicated renal impairment PK study be performed for drugs that are intended for patients with kidney disease, and that if the drug is mainly eliminated by the kidneys. Now, small proteins and peptides may undergo glomerular filtration, so a dedicated renal impairment PK study may be warranted in this situation. These types of studies are not needed if the drug is going to be a single administration only, if it's a topical drug, or gaseous or volatile. And then larger proteins with a kilodalton greater than 16.9 tend to undergo other catabolic processes for elimination. So a dedicated renal impairment PK study is not needed. In terms of drug development timeline, it's always good practice to conduct a renal impairment PK study once the ADME data is available and before entering into phase 2 and phase 3 clinical trials, because the findings from this study could really impact the inclusion-exclusion criteria for the patient trials. Because of the widespread availability of eGFR into current clinical practices and the limitations of creatinine clearance, the FDA recommends to use eGFR for these types of studies. Now, they don't provide a specific formula, but in the past, the MDRD had been widely applied, yet the CKD-EPI equation is gaining in popularity. We haven't seen widespread use of the combined formula yet, but given the endorsements from the kidney associations, this may be changing soon. And then again, it's important to de-index the eGFR and provide the values as mils per min. The classification shown in the table of controls, milds, moderates, and then severely impaired and kidney failure not on dialysis really should be used for these renal impairment PK studies and not for diagnosing or classifying the disease itself. As the name implies, a full design covers the full spectrum of renal impairment. And this type of study design is typically recommended for drugs that are mainly excreted by the kidneys. A reduced design incorporates the worst case scenario, so you have the control group compared to severely renal impaired patients or kidney failure patients not on dialysis. This type of reduced model is recommended when the drug is excreted through other routes. The recent FDA guidance also now recommends to include a sample size justification. So while previous versions of the guidance didn't specify the sample size, and typically 6 to 8 patients were enrolled per cohort, now it's important to do the sample size calculation. This will give more confidence in the outcome of the studies, but also note it will have a dramatic effect on study timelines, the number of sites needed, and potentially study costs as well. Our biostatisticians calculated the recommended sample size required to achieve 80% power based on the standard deviation and variation. And in the table, you see the N ranges anywhere from 7 to 24 patients per cohort. In most cases, a single dose is administered. However, multiple doses may be needed, especially if the drug is dose or time dependent. In this case, a lower dose or less frequent dosing may be administered in order to avoid buildup of the drug for safety purposes. And if safe to do so, we may want to consider a loading dose to achieve steady state faster. PK samples are collected and measured out to 3 to 4 times the half-life, and then major and active metabolites should be measured as well. For sample collection, analyze unbound drug if plasma protein is greater than 90%. Here, a limited number of unbound samples can be measured if the binding is not concentration or time dependent. So we would suggest then a sample at baseline and then again at the Tmax. A dialysis study is recommended when the drug is likely to be used by patients undergoing dialysis or renal replacement therapy. This would also be considered important if the drug is not too tightly bound to plasma proteins. And there's 3 key questions that a renal replacement PK study should address. And that is dose adjustment needed due to the dialysis procedure? If so, what is the dosage? And what is the timing relative to the dialysis procedure? In the US, intermittent hemodialysis is the most common dialytic modality and typically recommended for these PK studies. In this case, each participant should receive the study drug both on and off the dialysis. And then continuous renal replacement therapy is typically used in the ICU setting for critically ill patients. If this type of dialysis modality is being thought, then the FDA recommends to reach out for input on the study design. For data analysis, it's important to measure and collect plasma and urine PK samples. The key PK parameters are listed here for the parent metabolites, as well as renal replacement therapy PK measurements. Pharmacodynamic samples can also be measured when appropriate. For example, you could consider measuring uric acid for a gout drug in development. Now, an alternative approach to running a dedicated renal impairment PK study is to model the data. eGFR can be determined in a phase 1 study and phase 2 studies, and then modeling and simulation can be applied to get an understanding of how this could potentially affect patients going into your phase 3 trial. In addition, POP-PK approach can be used to collect information in a phase 2 and phase 3 trial, as long as there are sufficient numbers of patients with renal disease and appropriate PK sample collection. There are several design options and approaches for these renal impairment PK studies. A parallel study design recruits and enrolls subjects of each renal function category and the match controls all at the same time. So the advantage of this is that you could potentially reduce the study duration because all of the conduct is happening at the same time. A staggered design involves 2 parts typically. In the first part, we recruit and enroll patients from one renal category, for example, the severes and their match controls. And then after data analysis and review, the part 2 could continue in either mild or moderate patients with their match controls. The advantage of this approach is that you might not need to move on to part 2, and then you end up with a reduced design, and there are potential cost savings there. And then a sequential design is typically used with the 2 periods for a renal replacement therapy type study, where the patient is given the study drug on and off the dialysis day. The more conservative approach is to start off in the mild renally impaired group, gain information about drug safety, the drug PK, and then move into the moderates and then the severes. Whereas a more direct approach is to start off with the severes and the match controls, as this may be the most affected population, and then move towards the moderates or mild groups if warranted. Now, the approach will depend on the study drug safety profile and whether you are planning to do a full or reduced study design. Renal impairment patients often present with multiple comorbidities like obesity, diabetes, cardiovascular disease, hypertension. So the IE criteria is a little bit different typically than a standard phase 1 type study. And here are some key recommended IE criteria that we found works best for these studies based on input from our experienced site investigators. So typically we suggest to enroll patients up to 85 years of age with a BMI up to 40. If possible, the protocol should avoid any stability or QT criteria, and to allow for repeat labs for minor metabolic and electrolyte abnormalities. The healthy control subjects are matched based on demographic and anthropometric measures like age, sex, body weight, and BMI. We typically recommend the age be plus or minus 10 years, BMI plus or minus 10%, for example. There are several strategies for matching. You could do a 1-to-1 match, and the advantage here is that you can enroll the patients and the controls all at the same time. This could become challenging, however, if a patient has a very unique characteristic. It may be more difficult to find that matched control. There's also mean matching, and that could potentially reduce the number of healthy subjects for this study. However, you have to wait until all of the patients are enrolled to be able to calculate that mean and find the control subjects. The control subjects should also reflect the key demographic of the intended population. So for example, if you're developing an Alzheimer's drug, then you want the healthy control group to be more in the older adult range. And while it's not necessary, it's always good practice to enroll the healthy subjects at the same site as the patients because they would be exposed to the same environmental conditions as well as study procedures. As I mentioned, these types of studies are multi-site studies, and typically we look at 2 to 4 sites per study. And the number of sites really depends on the study design itself, whether it's going to be a full or reduced study. Also, the number of severe patients, they could be rate limiting in terms of enrollment into a type of renal impairment PK study. The inclusion-exclusion criteria as well. For example, if there's any genotyping needed, we may need to look into several sites to help identify these specific patients. And then it also depends on the sponsor and their timeline and budget. In terms of specialized sites, Celerion has a robust site network across the US and Europe to run both renal and hepatic impairment PK studies. And across all of our site networks, we have access to more than 3,000 participants and have managed more than 37 studies. Celerion scientists design studies that optimize the number of subjects, samples, and assessments to meet the needs of the study. The same team of scientists apply knowledge of the drug across all studies when performing conduct and analysis. This allows Celerion to create efficient and cost-effective packages for studies that form the basis of specific label claims in drug applications. Our global clinical development project managers have identified 4 unique tasks to successfully executing these renal impairment PK studies. They include ensuring that the healthy control subjects are matched to patients according to the protocol-defined parameters, carefully reviewing concomitant medications for these patients, as they are often on many different types of medications due to the disease. Also tracking enrollment to ensure the appropriate number of subjects are enrolled in each cohort, and then ensuring patients are assigned to the correct impairment cohort as well. So with these, enrolling a subject that is not needed for an analysis can be a very expensive mistake. And that's why you want a knowledgeable team to manage these types of studies. Now, before we conclude, I'd like to review a case study example. This is an older example, but I like it because it highlights several of the design elements and considerations that we had discussed today. So this was a study with gefapixant, which is a drug being developed for chronic cough. The ADME results did indicate that it was mainly eliminated by the kidneys. And then considering the intended population, chronic cough is quite prevalent in older adults. So a full type study was recommended. This was a single dose, 2-part study involving patients with moderate, severe impairment and kidney failure patients as well. The inclusion criteria include those up to 80 years of age with a BMI up to 40, and then the MDRD was used to estimate GFR. The controls were mean matched by age and weight. And this 2-part study, part 1 enrolled the moderates and then the severes. And then in part 2, we had the end-stage renal disease patients, both on and off dialysis, with the mean matched controls for all 3 cohorts of kidney dysfunction. And 2 sites were used. Now, in the graph, you'll note that there was higher exposure with the moderates, the severes, and the end-stage renal disease patients compared to the healthy controls. And the exposure response analysis revealed that there was a relationship between renal function. The area under the curve increased with decreasing renal function. The results also revealed reduced renal clearance and that the drug itself was affected by hemodialysis. Overall, it was well tolerated, and the results from this study was used to inform their phase 3 clinical trial design. Now, to sum up, the application of the race-free EGFR formulas are recommended by the kidney disease associations, as well as the combined creatinine and cystatin C formulas. For renal impairment PK studies, in the past, we've seen the MDRD be applied. However, the CKD EPI formula is also gaining in popularity. And it'll be interesting to see the uptake of this combined formula as the industry progresses. And the FDA also recommends to power these studies and de-index by the individual's body surface area. The optimal study design for a renal impairment PK study really depends on the drug safety, the drug PK profile, as well as the intended patient population. And Celerion has a dedicated, knowledgeable, and experienced team for smooth conduct of these renal impairment PK studies. I'd like to thank you for your attention, and please do check out our resource page on our website if you'd like to learn more about renal impairment PK studies. Thank you.